We only got to number 3 of the AP practice exam about the fur color. So what we're going to do right now is go over 4, 5, and 6 of the number 3. So let's check it out. So question number 4 talks about this pine wood nematode. That's a type of worm. And it's a eukaryote, meaning it has a nucleus, and it infects pine trees, feeds on the cells surrounding the transport system, and ultimately kills the trees. Just a reminder, the transport system of a tree is composed of the xylem and the phloem. The xylem carries the water, and the phloem carries the sugar. I think I mentioned in class sugar. You can think of this like maple syrup, right? You tap into a tree, you put a hole in the bark, and you can get the phloem coming down, and that's the sugar, that's the flavor you get from the maple syrup. And it ultimately kills the trees, it says. I'm not sure what percentage, but it says it's deadly. So they're infected with they're infected when nematode-carrying beetles. So it's really the beetles that infect the tree, and it kind of inject these nematodes into them. It's not the nematodes flying around, it's the beetles. So you got to worry about the beetles first. So the beetles feed off the trees, inject the nematode into the trees when they bite through the bark. And once infected, the pine trees increase production of chemicals that serves as a defense mechanism by negatively affecting the nematodes, so going after the nematodes once they get in there. So they've looked into this and found these nematodes have this special symbiotic bacteria. So this would be anything symbiotic means it's beneficial for both. So both the bacteria and the nematode benefit from this relationship. So these nematodes have a special bacteria that gives them a defense against the pine tree. So it's fighting back against these chemicals that the pine tree is throwing at them. So to investigate how bacteria play the role and how well the nematodes survive in there, they pre-treated nematodes with antibiotics and then exposed them to one of these defensive chemicals, this alpha-pinene. So first you got to describe the relationship between the parasite and the host. And remember, a parasite is going to be taking materials, resources from the host and not giving anything back. That means making it harder for the host to live. It's not mutually beneficial. It's not symbiotic. In this relationship, both the bacteria and the nematode benefit. So for A, you can write something to the effect of what I have here. Using something from that host, space, water, habitat, and harming the host in the process, making it less healthy. They don't always die from the parasite, but they don't thrive. Letter B, or the next question, explain how making this enzyme is beneficial to the bacteria species living within the nematodes. So if you want to fight back against this defensive chemical, you want to get rid of it. And that apinine digesting enzyme will help that nematode survive because it'll take away some of those chemicals that are making its life difficult. So, something to the effect of what you see here. Remember, the nematode wants to live and, have that nice, cozy environment where there's food around and it's not having to be chased by other the worms, they're at risk for getting eaten out in the world, but if they're inside of a tree, it's much better for them. The next question, predict the effect of an antibiotic treatment on the mortality rate of the nematodes exposed to A. piney. Now, you would imagine you get rid of that symbiotic bacteria, that's going to leave that nematode much more exposed to getting killed. So, reduce that nematode's ability to counteract the tree's defensive chemicals. So the mortality rate would increase. Because it just can't fight back against the tree's defenses as well. And the last question, provide reasoning to justify your prediction. So I kind of went a little too far. Sometimes you just got to be careful when you're doing these. Be really careful. Don't put more information than you need to. If it just asks you to predict the effect, oh, the effect is more are going to die. I kind of added too much of the explanation to mine. So here's an explanation of where it should be in answer area D. They would kill that symbiotic bacteria, and since that bacteria degrades the defensive chemicals, having more of that defensive chemical around is going to make it harder for the nematode and probably would die. So that's an explanation for question number 4. Let's move on to question number 5. We have a cross section of a chloroplast showing the stroma, which is like the cytoplasm. So the stroma, that's like cytoplasm. It's just the fluid that is in the chloroplast. And the grana are stacks of thylakoids. Those are compartments inside the stroma. We see 2 membranes, one on the outside, one on the inside, and then we see actually a third membrane system inside. So the major process, singular, and I messed up on mine, the major process that takes place inside this eukaryotic organelle, which is a chloroplast, is you take CO2 from out here and you're making sugar. Okay, you're what's called carbon fixation. You're fixing carbon from the atmosphere into sugar, some kind of chemical. So again, I kind of went overboard. So this is what you should have here, and that's 1 point. CO2 and water fix carbon dioxide into sugar. Let me put that in my diagram. Or I had that. Add water. And you also need some kind of light source. It can be natural or artificial. So I just kind of annotated that. So CO2 and water fixing carbon, that's worth 1 point. The second question, explain the function of the structure labeled with an X. So that would be the outer membrane. So the outer membrane allows the chloroplast to have an area to separate these reactions to make them more efficient. That's all you got to put for that. And then with on the template and the space provided, which is on the second page, it wants you to represent the location. So it's all about where in this diagram the CF, which is carbon fixation, and the electron transport proteins. Remember, transport chain in the light reactions that happen in here in the thylakoids. So it just wants you to, it's asking you if testing you if you know where those take place. So there might be some times where it's going to ask you to write on a diagram. So just make sure, don't feel uncomfortable doing that. All right, so carbon fixation. Go to our diagram. You can have it anywhere in the stroma. Carbon fixation happens. Remember, this is in the stroma, this is where you have the Calvin cycle. Oops, Calvin cycle. That's where you have CO2 coming in, you have sugar going out. That's also the light independent. Not sure. Yeah, light independent reactions. So that's in the cytoplasm. So 1 CF anywhere in the spots that I have will work. You don't have to have it multiple places. And then the electron transport chain, or I think I messed up, electron transport proteins, ETP is what they want you to put there. It's where you have these membranes for the thylakoid. And these would be the light dependent reactions. So any thylakoid you can write ETP on, that will work. And the last question, explain the shape and the stacking of the thylakoids, how it contributes to the rate of carbon fixation. What you're seeing here, this really, see how flat and spread out these thylakoids are? There's a lot of surface area, and we've talked a lot about surface area. We had that lab on surface area with the cubes, the pink cubes. So the more surface area, the better. And the way those are flattened out, that's maximizing the surface area. And that surface area is where you have those electron transport proteins and the electron transport chain. The more chains you have, the more ATP and FADH2 you can make. So it's a huge advantage to increase your surface area. So that's stacking. So this is the answer. I believe, I'm sorry, I can't remember how many points this is. I forgot to write it down. I believe it's 2. But the stacking increases the surface area, and that allows for more chlorophyll molecules to be exposed to light, more electron transport proteins. Yeah, this is where the chlorophyll is. It's in these thylakoids. So the chlorophyll that we pulled out, using our paper chromatography, that's where they're at in these thylakoids. Is here, okay, in thylakoid. Is here in the thylakoid membrane. Okay, more energy. More surface area, more energy. So make sure you have at least one here and one here, you're good to go. And I just to be helpful, just want to make a note of this. Surface area is just such a huge, giant concept in biology. Comes up all the time. I'm sure this will come up a couple times on the exam. And here's some examples. Leaf surface area, more light. You don't see round ball-shaped leaves. They're always flat. So you have surface area to catch the light and to exchange gases. The root hairs on plants, on the roots of plants, they have these hairs that come off. And the roots themselves are designed. They're not really thick usually. You do have tap roots, but you have many little pieces coming off those tap roots called root hairs that increase that surface area. In our body, in the alveoli, our lungs, you got tons of surface area so we can exchange oxygen and carbon dioxide. In our intestines, the other major example that we talk about, you have these villi that poke out from the sides of your small intestine, and then microvilli on top of them, just building and building more surface area to absorb as many nutrients as you can. Without those, you'd have to have double, triple, quadruple the length of intestine, and we don't have time for that. That takes a lot of energy to carry around more intestine. So increase the surface area. Skin surface. You see animals that are desert adapted tend to have more skin surface area. Warm climate animals, so they can get rid of heat. And then animals that live in the cold, you don't see long arms and long legs. in animals that live in cold environments usually, or in the ocean. You see a reduction in skin surface. Gills in fish, tons of surface area along those capillaries in the gills to facilitate gas exchange. And in your body, the tiny, teeny, tiny capillaries that go everywhere, they're teeny tiny so they can exchange nutrients and gases more easily. All right, last question. We have some fruit fly. And we do use these a lot for genetic studies. So we're looking at two. It has 2 of each of 4 different chromosomes. They have a sex chromosome and 3 autosomes. I guess they are labeled 2, 3, and 4. And we're looking at genetic crosses to look at X-linked recessive trait encoded by a single gene. So I wrote a note here. This is similar to hemophilia that we looked at before. Remember, if you have an X-linked recessive trait, females need 2 copies of that recessive trait in order to get it. Men need only 1, and since men have only 1 X, they're more likely to have it, to get that trait. So this is our data table, the crosses they did, unaffected, affected, affected, unaffected. You know, they just did all the different combinations. And they're asking you first to, what are the genotypes of the male and female flies used in cross 2? So if the female is affected, that means they must have 2 lowercase. And they gave me R to use in this example, but you could use any letter. Just make sure it's lowercase, like a little h for hemophilia, for example, versus capital. So if you're affected female, it's got to be 2 lowercase, 2 recessive alleles. And unaffected male, it has to be a capital R. So here's your answer right there. Which cross identify in the cross in which the female parent was most likely heterozygous? So that would be cross number 3. Because if the female is unaffected and the male does not have the disease, but look, they have affected offspring. And we know from our Punnett square here, this is again the same Punnett square we would draw for hemophilia. Non-affected mother, non-affected father, but look, they have affected offspring. 1 out of 4 or 25%, which is what we thankfully see. It's amazing how this worked out so perfect. I don't think this is a real study. I think this is one of those fake studies AP likes to, with made up data. Oh yeah, it's not. Yeah, there's no reference or citation. So yeah, AP created all this data for us to make it nice and easy. So cross number 3, female. But remember they have the offspring who are affected, just like the Tsar and Tsarina. This is the Tsar. This is the Tsarina. One of my favorite lessons, recovering the Romanovs. So going on to, and those are both 1 point. The researchers thought or hypothesized that crossing an unaffected female and an affected male would result in 0% chance of the production of affected male offspring. Evaluate the validity of that. So let's just quickly look at this. Let's try this out. Any unaffected female, the key is the male is affected, right? So the male would be that. And we'll just say the female is homozygous dominant. And in this case, everybody would get a capital R, right, from the female. But, and you would just have carrier, these would be carriers. But if the female, kind of run out of space here. If the female is heterozygous, then she can give that to this male and have an affected male offspring. So if the female is heterozygous and no matter what about the father, there can be an affected offspring there. So it's not supported. The female could be heterozygous and carry a recessive allele that could be passed on to the male offspring. Now the last one is how the results exclude the possibility that the trait is encoded by the mitochondrial gene. And we know it told us in the question that it's X-linked recessive. So we know that that is a chromosome located in the cell's nucleus. A mitochondrial gene would not be on the chromosome in the cell's nucleus, and it would only be inheritable from the mother. So in this case, father could pass on the trait if there's a recessive gene is on his X chromosome. And this kind of is confusing because we did talk about mitochondrial DNA in the Romanov's activity, but that was only to get a DNA match. It wasn't for hemophilia at all. We were just trying to match the DNA. We could have. The researchers could have used nuclear DNA for their comparisons, but nuclear DNA tends to degrade more and be less likely to still be good, 80, 100 years later. I guess it was about 80 years later when they tested it. So mitochondrial DNA is used because it's well understood, very tough. It doesn't break down very easily. Fairly easy to extract, and it can be easily matched with current relatives. So that's the final. Now you have all of the corrections, all the keys for that second test, and hopefully it helped you.